Hi everyone, welcome back. This week we're going to be exploring two new topics. We're going to be talking about tubulin, which is a very important protein. We'll be talking about what it is, where it's found, and what it does. And then we'll also be talking about a medication called Oncovin. First, let's start off our exploration this week with tubulin. Tubulin is a protein that's critical to cell division, and it's present in two forms, alpha tubulin and beta tubulin. Now, each of these forms of tubulin has a slightly different structure, and it does something slightly different. So alpha tubulin has an aspartate instead of a lysine in the 254th amino acid position. Beta tubulin has a lysine instead of an aspartate in the 254th amino acid position. Now you may be asking yourself, what is so important about the 254th amino acid position? There's many amino acids in this molecule. Why is that one so important? And that's a great question. The reason it's so important is because in alpha tubulin, the 254th amino acid serves as the ideal site for hydrolysis at the E site. And in beta tubulin, this amino acid strengthens the interaction with the GTP phosphate group in the N site at that location. And these forms of tubulin polymerize to form microtubules during mitosis, which is what you see on the right-hand side of your screen. Each microtubule is made of 13 linear protofilaments with alternating alpha and beta tubulin molecules. These microtubules form the mitotic spindle, which helps move and separate chromosomes during chromosomal segregation. Now, it's important that these chromosomes move apart because if they don't, then you can end up with non-disjunction and mutations that occur. The tubulin does this by attaching two chromosomes at the kinetochore. Over on the right-hand side of the screen, you can see here we have a microtubule. We have the alternating alpha and beta tubulin structure that we talked about. We have the 13 protofilaments, and you can see that on this cross-section, microtubules are hollow. Now that we've talked about tubulin, let's talk about oncovin, which is also known as vincristin sulfate. Vincristin is an alkaloid drug that's derived from the vinca plant, which is also known as Madagascar periwinkle. And you can see Madagascar periwinkle and what the plant looks like on the right-hand side of the screen. Vincristin was discovered along with a different alkaloid called vinblastin in the 1950s when researchers were exploring the use of vinca-derived alkaloids as treatment for diabetes. Pure vincristin was isolated in 1961, which is when it was found to treat induced leukemia in mice during clinical trials. Vincristin was approved for use by the U.S. Food and Drug Administration in 1963 under the trade name Oncovin. It was marketed by the Eli Lilly company, and Eli Lilly still holds many of the Oncovin patents today. Vincristin is indicated for the treatment of acute lymphoblastic leukemia and acute lymphocytic leukemia as monotherapy. However, it's typically used in combination with other chemotherapy drugs to treat such conditions such as Hodgkin's lymphoma, non-Hodgkin's lymphoma, rhabdomyosarcoma, neuroblastoma, and Wilms tumor. So Oncovin, or vincristin, is comprised of two multi-ringed units of vindolin and anthine, and is cell cycle phase specific. It works by binding to the microtubular proteins of the mitotic spindle, which leads to crystallization of the microtubule. What this does is this inhibits microtubule formation in the mitotic spindle, which effectively halts cell division during metaphase. And Oncovin targets both alpha and beta-tubulin chains that we saw earlier. It may also help to function by interfering with metabolism, specifically metabolism of amino acids, cyclic AMP, and glutathione, as well as calmodulin-dependent calcium transport ATPase activity, cellular respiration, nucleic acid synthesis, and lipid biosynthesis. Vincristine, however, does have a significant limitation in that it does not penetrate the blood-brain barrier well, and it also does not cross over into the cerebrospinal fluid. So central nervous disease progression has been noted in individuals who are on monotherapy with Oncovin. So that is why it's typically used with other medications that have better penetration of the central nervous system. Metabolism of vincristine in the body is mediated by hepatic chromosome P450 in the CYP3A family. What's great about Oncovin is within 15 to 30 minutes after administration, 90% of the medication is transitioned from the blood into the tissues. Over on the right-hand side, you can see the double ring structure of Oncovin, or vincristine. Now that we've talked about the benefits of Oncovin, what are the side effects? Side effects, as with any chemotherapy drug, come along with Oncovin. The most common is hair loss. However, there can also be significant neuromuscular side effects such as paresthesias, difficulty walking, changes in gait, loss of deep tendon reflexes, muscle wasting, and hearing and visual loss. It can also come with hematologic consequences such as leukopenia, anemia, and thrombocytopenia, which again is not uncommon with chemotherapy medications. Gastrointestinal symptoms following Oncovin administration can include constipation, abdominal pain, weight loss, nausea, vomiting, oral ulcerations, diarrhea, paralytic ileus, intestinal necrosis, intestinal perforations, and anorexia. And a side note, this should not be administered to pregnant women or nursing mothers. Here are my references for this week, and thank you so much for spending a few minutes with me to talk about tubulin and Oncovin and how the two are related.